by giving a brief overview of the background of a project and its specific aims. I'll follow up by talking about the latest updates and give a live demonstration of a Google Chrome extension we've been developing. And finally, wrap up the presentation by talking about the next steps and how this could potentially be implemented in the BioCaddy web portal. So as we all know, there's a large amount of biological data stored across multiple repositories online. And the DataMed indexes many of these, such as the Gene Expression Omnibus, NERSA, Links Data Portal, and PDB, and many other repositories which contain a large wealth of transcriptomic, genomic, proteomic, and other types of biological information. On the other hand, there is a wide variety of computational tools which can operate on these datasets to extract valuable biological knowledge. Many of these are indexed in repositories such as Aztec.data, developed by BD2KCCC, or omic tools. And these include gene expression enrichment analysis, visualization tools, and many other different tools. So the use of the computational tools on the biomedical datasets is a key step in extracting knowledge from them, which can only lead to increased understanding of biological systems. However, none of the aforementioned repositories currently provide methods to systematically associate all the datasets stored within them to the computational tools which can operate on them to extract knowledge. So the key aim of the project is to promote the association between computational tools and the biomedical datasets which can be analyzed by them by introducing a new type of digital object we named the canned analysis. So to achieve this, we aim to enrich DataMed with a series of interfaces which will increase the accessibility, findability, and shareability of this new type of object, which contains results of analyses run by the tools on the dataset. So the key question we asked ourselves when defining this project is how to define a simple but effective solution to the issue of connecting datasets to the tools which can operate on them. And the model we proposed to address this issue is a new type of digital object we named the canned analysis. So essentially, the canned analysis is defined by three key elements. The first element is a link to a biomedical dataset which is stored online on a biomedical repository, can be accessed through a URL, and can be downloaded by users and used to perform analyses. The second element is a link to a computational tool which can operate on the dataset to extract biological knowledge. Ideally, this tool exists online, has a home page which can be accessed through a URL, and produces as a result another web page which contains all the results of the analysis run on the dataset. The third element which defines the canned analysis is an element we named the canned analysis URL, which is a hard-coded link to an external web page which contains the results of the analysis run by the tool on the dataset. And again, this should be easily accessible by users simply by going to the specific URL. The canned analysis can also be further enriched by adding a description, which would allow users to better understand the purpose and aims behind it, and any number of customizable metadata tags to further annotate it accurately. So to give you a better feel of what a canned analysis is, I will give a few examples of these objects. So the first example of a canned analysis could be an enrichment analysis on the top most overexpressed genes in a knockdown perturbation of a transcriptomics dataset. So for example, we could take a geodataset, in this case GDS 4054, which is an analysis of microRNA 221-222 knockdown on fulvestrant-resistant MCF7 breast cancer cells, which is accessible in the URL shown here. The second element is a link to a computational tool which can operate on this dataset. In this case, the computational tool is Enricher, which has been developed by the Mayan lab, which allows users to calculate the enrichment of gene sets over a large number of different biological pathways and terms. The third element which defines the canned analysis is the canned analysis URL, which in this case is a web page which is hosted on the Enricher website. And upon clicking on this web page, on this link, the users are directed to a web page which is shown in the bottom right corner of this slide. And in this case, the user will be able to explore the enrichment results of the specified analysis. The description of the analysis could be, for example, an enrichment analysis of the top 500 most overexpressed genes in a microRNA 221 knockdown perturbation MCF7 cells. And the metadata which is associated to the analysis could be informative about gene set which has been generated to perform the analysis, So for example, whether it's upregulated or downregulated genes, the number of genes inside it, and for example, the perturbation type. Another example of a canned analysis could be an interactive heat map visualization of transcriptomics data sets. So in this case, we have another GEO data set, GAC 44875, which is analysis of the effect of a long non-coding RNA on the expression of a transcriptional factor FOXA2. The tool in this case would be Clustergrammer, which has also been developed by our lab and allows users to create interactive heat map visualizations of any kind of tabular data. The canned analysis URL in this case, shown in the bottom, is a link to a page which is stored on the Clustergrammer website. And upon clicking on it, the users are directed to a web page, as shown in the bottom right corner, which contains an interactive visualization of the actual data. It allows users to explore the clusters of co-expressed genes, identify enriched terms, identify similar samples, and so on. A description could be interactive heat map visualization of the top 1,000 most variable genes in the data set, and the metadata associated to this analysis could be the number of genes, how the genes were ranked, or the method which was used to normalize the data. So the final example, just to make a point that this could potentially move beyond transcriptomics to accommodate any other number of data types and tools, is an example of a structure visualization tool using a data set from PDB. In this case, the data set would be the 1STP data set, which contains structural information about binding to streptavidin. The tool could be the PDB Ligand Explorer, which is available on a URL on the official PDB website. Again, the canned analysis URL is a link of a web page which is stored on the PDB website. And upon clicking on it, the user will be redirected to a web page which contains a 3D visualization, in this case, of the binding between biotin and streptavidin. So briefly, essentially canned analyses will be designed to support multiple different data set types, such as transcriptomics, genomics, epigenomics, and any other types, as well as multiple types of computational tools, which could be enrichment analysis tools, visualization tools, but ideally any other number of computational tools. So the canned analysis is a series of key properties which we think will make it useful to address the issue of connecting data sets and tools. So first of all, the structure of its metadata is independent of both dataset type and tool type. This allows for interoperability between computational tools and data sets coming from non-cooperating platforms, and also allows accurate annotation of each digital object, independently of the data and tool which has been used to generate them. Second, all the information about canned analyses, including metadata and keys, is stored within a relatively simple relational database and can be easily retrieved by performing database queries. Finally, the object exists entirely online and can be easily shared between users in the scientific community by means of a URL, which can also be embedded in websites as an icon. So one of the key questions would be, why would I be interested in saving my results as forms of canned analyses? So the idea behind this is that the Datasets to Tool platform will allow to greatly increase the accessibility, findability, and shareability of analyses which are stored as under these digital objects. So to achieve this, we aim to develop interfaces which will allow users to easily find canned analyses associated to relevant data sets directly from within the dataset web pages, to rapidly access the results, to view and download the metadata associated to the analyses, and to share these with other users. So the first implementation of these interfaces will be performed by using a Chrome extension, which will be available for free on the Chrome Web Store and can be easily installed with a few clicks in the form of a plugin on the Chrome browser. We aim to make this active, of course, in DataMed and extend this to the Links data portal and GEO. If the interface is successful and we receive good feedback on it, potentially moving forward, we aim to make this native to the DataMed web portal, which I will discuss at the end of the presentation. So I will now give a brief demonstration of the functionalities added by the Chrome extension on DataMed. So opening up the DataMed web portal, let's say somebody's interested in finding more about MCF7 cells. So he would type in MCF7 in the search bar, And currently, the database only contains dataset analyses which are stored to geo datasets. So I'll now click on the geo filter. But moving forward, as we populate the database, we aim to extend this to other repositories as well. So the functionality added by the datasets to tool extension is shown as these toolbars below some of the search results. So the structure is pretty simple. We have the logo of the project name, and then a number of icons which represent different computational tools which have been used to analyze the corresponding data set. By hovering over the icon, we can see the name of a tool, the number of analyses which are associated to the tool and dataset pair, and a brief description of a tool. In this case, you might recognize Enricher, which again is an intuitive web-based geneless enrichment analysis tool which allows users to calculate enrichment of gene sets over a large number of different pathways and terms. So upon clicking on one of these icons, the user triggers a drop-down menu containing a table where every single row represents a different canned analysis. The first column contains links to the canned analysis URLs. So upon clicking on one of these links, the user is redirected to an external web page which contains the results of the analysis. So in this case, the user can explore the enrichment results of the specified gene set. The second column contains a brief description of the data set of the analysis. In this case, this is the analysis I showed as the first example in the presentation. So it's an enrichment analysis of the top 500 most overexpressed genes in a microRNA 221 knockdown experiment. The third column allows users to explore metadata associated to the analysis. In this case, we can see the number of genes, the direction of a gene set, the perturbation type, and other relevant information. The metadata can also be downloaded in a variety of formats, such as text, in the form of a table, or JSON files, which can be seen here. Lastly, if users want to share the analysis, they can click on the last button, and it currently allows users to copy the URL to the canned analysis, which can be then pasted in to access the analysis results or shared with other users. And also provides a brief snippet of HTML code, which allows users to embed the link as an icon in websites. If users want to search specific analyses, they can use the search bar. For example, we can look for only perturbations of microRNA 221. And finally, if the users want to have additional information about the computational tools, they can click on the information icon beside the tool name. This will open another menu containing a brief tool description, links to the tool's homepage, eventual publications. And we aim to extend this by adding more detailed descriptions and, if available, video tutorials or similar. So this wraps it up for the main functionalities of the Google Chrome extension. The same interface is also available on the dataset landing pages. So in this case, the Chrome extension adds a tab named Canned Analyses, which contains a very similar interface. In this case, instead of having a toolbar, we have a table where every row represents a different tool which has been used to analyze the data set. The first column contains the icon and the tool name. By clicking on it, we are redirected to the tool's homepage if the user wants more information about the tool. Again, we have a brief tool description and the number of canned analyses which are associated to the tool and dataset pair. By clicking on the plus button, we are directed to another table which is the same as the table I showed before. So we have information about the tool's link to the links to the canned analyses, a brief description of the analysis, metadata, and sharing functionalities. So this wraps it up for the actual functionalities and implementation of the Chrome extension in DataMed. This extension also currently supports Geo and the Links data portal. So now go back to a presentation and present a couple more slides, which will wrap up for today.
Dennis, I have a question. If
yep.
you go back to your demo.
Of course.
So when we had the search results, if you go to the previous page.
Yep.
Yeah, here. So when the drop down happens, you have multiple rows, right? So why do we have multiple rows? Because the information seems to be the same.
So,
them.
right. So actually, every single row represents a different analysis. In this case, they're all different. So the first row has the overexpressed genes.
Oh, I see.
Second row has the underexpressed genes. And the third row has the combined underexpressed and overexpressed genes. So the first three represent the same gene signature, but the first analysis only takes the top genes, the second takes the bottom genes, and the third one takes the combined genes. And this is for microRNA 221. Then we have the same thing for microRNA 222. Does this answer your question?
Yeah, yeah.
Okay.
So I just wonder if we can somehow make that obvious. Because
Yeah. So these descriptions I generated computationally. They're more of a placeholder, I would say, for now. But the actual description is up to users to insert appropriately. But I'll write it down. You're right. It could be made more intuitive.
Because I mean, when I was looking at it, I just probably looked at the first line, and I was like, oh, all of them look very similar. But you're right.
Yeah, no, they are different, but it's a good point. Thank you. All right, so moving forward, we hope that these interfaces could become part of the DataMed web portal. And in terms of actual contribution from the BioCaddy development team, we would require very limited work. So the databases containing information about the canned analyses, the metadata, and the links are hosted locally at Mount Sinai facilities. And the interfaces will be developed here. The code will be open access, and if this moves forward, this would also support multiple browsers aside from Chrome, such as Safari and Firefox. In terms of future directions, moving forward, one of our next interests would be to start populating the database with canned analyses which are submitted by users. So we currently have a template of an Excel table which would allow to submit analyses to our database. It would require users to specify datasets, a tool, the canned analysis URL, and a description of the analysis, and potentially any number of tags which are associated to the analysis. Once the information is inserted in the table, it can be then uploaded by means of an API to the database. And by using the dataset to tool interfaces, it will be available on the BioCaddy DataMed website and somehow return and made accessible to users in the scientific community. So wrapping up, a last slide. Ideally, these canned analyses, as I mentioned before, will not only be limited to transcriptomic datasets, but could potentially be expanded to support any number of data type and tool type. So for example, they could, an example of a canned analysis could be a visualization of any type of tabular data with a clustergram tool, or visualization of 3D structural data of proteins, ligands, and interactions. Of course, analysis of gene expression signatures, and potentially any other type of analysis run by tools on datasets. And in this regard, Kathleen's survey on the dataset and tool types on DataMed would be very useful. I think she'll talk about it briefly soon. So this wraps it up for the presentation, and I'm happy to take questions if you have any.
I think this is a great presentation, and it's actually very cool. My question is more from CDT team, if when we start integrate this, so I probably not completely understand how this. So right now it's a Google Chrome
yes.
extension. And when we try to implement at the DataMed, will it be still be an extension, or it actually will be implement into the DataMed?
So the extension currently exists as a JavaScript file. So
okay.
I think the actual implementation of the interfaces within the web portal would be quite simple, as in simply load this external JavaScript in the website. So
that will happen at the DataMed, right?
Yes.
Our team
That's
will
what.
do that. But the JavaScript will talk to sort of a database on your side, right? You will be responsible for maintenance, like a.
Yes, that's correct.
Okay. Yes. Okay. That makes it clear. Because I'm not very familiar with those Google Chrome extension, but yeah. No, I get it.
That's correct. So
Yeah.
all the. Yeah, sorry, Abby.
No, I just want to say that's we will be very happy if you guys adopt the JavaScript so people don't have to install the Chrome extension to get access to that.
Yes,
So
yeah.
And then if you want to host the database on your side, that's also a possibility. Like, we are happy to host it here, but if you want to have full control of the content and make sure that it's maintained and it's up, and you don't want to rely on us, that's also a possibility that we will transfer the database. But the problem would be to maintain it and extend it.
Mm-hmm.
So we prefer to have it here, but if you are interested in hosting it or adapting that database for long-term use and want to take on that, that's also a possibility.
Okay, I see. Yeah, we can talk to Jeff and others or Lucila. We see how I don't know if there's any thoughts about we want to make all the data manager related stuff all together as a thing, or we want to make it distribute rely on different services. Maybe something in the between. Maybe we can also maintain a local database, but we somehow have a mechanism to get updated from your guys in a defined frequency. So then we can still rely on you guys to maintaining it.
Yeah, I think one thing to do is to check whether, right now when we are doing the search or when we're looking at a page on DataMed, only if there is data in the database it will display something. Mm-hmm. So it can do some type of a check if the server is up and there is an analysis and the analysis is valid and it's been updated and the URLs are all pointing to the correct site, then display the information. If there is any failure by any of those points, just don't display.
Okay.
And that can be done through AJAX call or some calls that happen after the page loads, so it doesn't really slow down the process.
Mm-hmm. About the database updates, how frequently are you updating database? Is this something weekly, daily, monthly? Because if it's not that frequent, it's 3 months, a year, or in that case, maybe it may change our option to maintain another database in our site.
I think right now it's a rolling update, but what we can do is we have a six months release updates, and then the database structure should not change. So that's also a possibility to provide every 6 months an update and then push it to you if that works better.
Okay. Sounds good. Jeff, do you have any other questions or anyone?
No, I'd asked questions at the last webinar.
Okay.
We had everything.
Okay.
Any question from you guys? Okay. Sounds good.
Abby, this is Ron. This is a great example of the interactions and collaborations between the centers and BioCaddy. Have you brought this forward to the other centers groups and to the NIH staff monitoring the centers so that they're aware of this kind of outcome?
I'm sorry, I was I pressed on the hang up instead of
Oh.
the mute earlier.
Okay.
I just came back.
Okay. No, I'm sorry. I was just, maybe I was a little late in putting the question forward. I was just saying that this is an outstanding example of how the centers have interacted with and collaborated with BioCaddy to provide a significant value added to the user. And I was just wondering if you've highlighted this with the other centers and with the NIH staff that monitor links.
Yes, we're trying to communicate it so they know, and I guess we're gonna probably wait until we have a complete outcome, but we give our program officers a constant update of what's happening, and I think there is some excitement with this way that we thought of an idea of how we can potentially work together without, interfering with the workflow of BioCaddy. Because initially we tried to get a copy of the database and try to work in parallel, and that solution that we came up with, I think it's a good way for us to develop independently, and then the integration should be something that is going to be relatively painless. So I think that's going to be exciting, and we just have to make sure that we drive it all the way.
Yeah, I agree. I think this is going to be extremely important to the users ultimately. The fact that it's all possible to do it online suggests that this could be part of the commons or a service as a software as service in the commons for use in the cloud.
Yeah.
Yes, this is Philippe, and I think it was very nice work. Again, really congratulations. And I was interested in the kind of markup and metadata schema that you used for identifying, or in a way, just marking up the different tools that are available for analysis and the different canned queries, basically, or canned analysis, especially the metadata elements that you've shown on the UI. I was just wondering if you already had a JSON LD context file for those elements.
Yes, I think right now it's in a database schema, but it should be straightforward to create a JSON LD for it. So this is a great suggestion and something that we can follow up on.
Okay. And yes, just to follow up, but also because it echoes the question that Annie asked me about the kind of tags that you use for tagging both the data types and the tools. Is that available? Can you share that with the group? Just to, again, as part of the work to collect the kind of tags and annotation people use.
Yes, I think Dennis should be able to share the entire content of the database with you after the meeting. Yeah.
Yeah.
Fantastic. Yeah. Thank you very much. Cheers.
Are there any other questions for Dennis?
I don't think. Is there anything else? All right. So Kathleen.
Next slide. Is that visible?
Yes.
Yep.
Yes.
Okay, great. Okay, so I'll briefly be presenting to you kind of a supporting project that I completed in support of Dennis's work. Let's see. Basically, I surveyed the BioCaddy data types and tools so that we could have a more complete idea of what we have to work with to provide canned analyses. And so I reviewed the 12 different data categories and the 43 repositories within those categories, and created a PowerPoint presentation that contains over 500 slides just summarizing the content. For each repository, I recorded the file types used and the tools that are available to analyze the data. And so the tools include things like data analysis and visualization. I didn't include utilities like uploading or downloading data. I did include third-party tools in the survey, and most of the resources are freely accessible, although some of them I found do require an account to be created in order to access them. So that can kind of provide a challenge if we want to, integrate them in the canned analyses. So included in this survey are screenshots and URLs to provide easy access to each resource. And I found that most of the repositories do have useful tools available. Many of them didn't appear to include them. So I just have a couple of example slides that I'll share to give you a feel for what's included in the survey. First example slide, I have a tool from the Jackson Laboratory and the Mouse Phenome Database. This is a correlation matrix tool here showing some metabolic analysis. And basically, so I provide the general link to all of the tools for that database, and then on the left, the specific URL to this tool, and then a description of how it's used and what it's. As a second example, we have a tool from created by our lab called Drug Cell Line Browser, which is an interactive data visualization tool that allows the user to interact with drug and cancer cell line data. And so again, I provide the link to the tool and a description of how it works. And so basically, have this survey that just contains many dozens of these slides, and we're planning to use it to extend Dennis's work in canned analyses. And one consideration is how to address the tools that aren't necessarily going to be accessible or easily adaptable to canned analysis. So that's future work. And then integrating the survey content with what Dennis has already done. And then as requested on last week's call, we do plan to share this survey, and we might discuss, how that might best be done. So if there are any questions, I welcome them.
So, Kathleen, one quick question. Of the 43 repositories that you looked at, do you have an idea of how many of them have tools and how many don't?
Well, I'd say a rough estimate is it's a real minority that don't. Maybe around 10 of them didn't, maybe even fewer. But the good majority did have useful tools available. Any other questions? Okay, that might be it then.
So Xiaochen, do you have?
Yes, you can give me the presentation notes.
So. I think you can share from your screen now.
Okay. So I will talk briefly about this data extraction and visualizing project for BioCAD-linked supplements. So at the very beginning, the goal was to provide an easy and usable interface for researchers to quickly identify a data set of interest based on the geolocation associated with the data. And so we started looking at appropriate data set from DataMed. Unfortunately, there's a limited repository containing geolocation. So far, we only identified actually 2 out of the repository that collected by DataMed. However, this is not a limitation of this tool. Better utility will be achieved as DataMed expands, as we believe. So briefly, I will present the data we used, the clinicaltrial.gov and dbGaP for this project. So for clinicaltrial.gov data, we do have a lot of study with geolocation, and actually the number of geolocation of study available in the U.S. is about 800,000, and a total of 70,000 million number of subjects. There are overlaps, of course. So what are there in this data set? There are detailed description of each study, author, keyword, citation, sponsor, age, gender, for the subject. But this data doesn't have a race for the subject, doesn't have relationship between other study, but it does contain like which institution and where this study was conducted. So the location of this study do not have a coordinate, but it does have the institution name, and we use the Google Place API to get the coordinate back from to the geocoding. And this is done by Java. There's a limitation on how many queries we can issue. So we came up with a memory cache so we don't repeat the query that we have been issued before. So the good side is this provides a precise coordinate for to the street level, but as I said, it only supports up to 250K queries for free. Otherwise, we charge $1,000. So we did use some memory cache to solve this problem. And here's a distribution of location in the US for these studies. Later on, I will show a demo on how we visualize and quickly retrieve data set of interest. And another data set that contains geolocation is dbGaP. There's a limited number of studies that has geolocation. Here we only have 429 studies have, from the data map, and out of them there are only 345 contains geolocation, and the ones in the US is even less, so it has 234. The number of subjects is roughly around 500K. In this data, we have detailed description for each study. We have author, keyword, age, gender, relationship between other study, but it doesn't have race sponsor. The location in this data set is not fine-grained. It usually only contains like state or like a city level. It doesn't even contain like institution sometimes. So we can only roughly get the location here. So here's the distribution. As you can see, like US is like 38% of the origin of the study generation. And we developed the portal backend using Amazon EC2 hosted HTTP server on Apache, and we used Elasticsearch to support the query API and speed up using memory cache. And in the front end, we use HTML5 CSS jQuery. We use the leaflet map API and Mapbox for the metadata, and we use the cluster library as well. So we pass the input string and send the query to the backend search engine via Ajax, and we retrieve all the matching locations in a dynamic manner. And we show a progress bar indicating the progress. And the working progress is we try to integrate not only the geolocation of the study, but also some description on the data if the data is available. So the idea is to, if we have data, we convert into the OMAP CDM and throw some of the statistics to a list developed by Columbia University and show a composition of the data in addition to the metadata description. So here's like the visualization of this tool, and if you put in lung cancer and the stage 2, it will show the 717 studies on this map, and each circle, which is color-coded by its size, indicates the number of studies. If you move the mouse, it will show the number of subjects involved in the study. The legend actually indicates the number of subjects rather than the study involved in this location. If you left-click, and it will expand, and it will move to smaller and smaller location, showing finer and finer granularity up to the individual study. As you can see, we can see like individual study occurred in US, in California, and this can be utilized in DataMat as a widget. So you don't need to show it in the full size, so you can use this widget mode and specify the height and the width as wished and embed it into a web page. Now I'll show a quick demo. So here is the website. If I put in, say, then you see the progress bar and is finished. This is from clinicaltrial.gov and defaulted US. So if I mouse over, you will see the number of subjects in California. If I click, then you see expansion. If I keep on clicking, you will see fine-grained expansion. If I right-click, it will show the decomposition of Achilles. Of course, this is simulated. We don't have the data involved in this study, but we launched some Mimic3 data statistics to just visualize. So in the future, if we do have access to the underlying data, then we can present not only the geo distribution, but also some study and population distribution associated with the data. So that's it for the presentation. Thank you.
Are there any questions for Xiaochen?
So, Xiaochen, how quickly we can integrate this to data map? Are we going to integrate this to data map?
No, it can be integrated, but the limitation right now it only has two data repository contains geolocation data. Only dbGaP and the clinicaltrial.gov. Other data set doesn't have it. But if you want to search for those two, it's ready to work.
Maybe we can treat it as like those pilot projects, have a kind of demo
right
thing.
Right.
We had for some
So like a proof of
IC
principle,
dam.
right?
Yeah.
Okay.
I think you can get the location from other studies, but looking at the institution.
Mm-hmm.
So if you, for example, for GEO, you can get the location by the author's institution.
Uh-huh.
So the author information is there and the school is there.
I see.
For links, the data is associated with a center.
Uh-huh.
So the center has like a primary location.
I see.
And there's only like 6, centers.
I see.
But still, I think it's. I think there are other ways to get the location for some other data sets.
Okay. I will check with team on that. Yeah.
So, Xiaochen, the data, the geolocation that you have is for the subjects in a study, though, right? It's not for where the study is conducted, right?
No, I think it's where the study was done, not the subject.
Oh, okay.
Yeah, for the clinicaltrial.gov, I only have the institution that initiated the study. I don't have the distribution of the subjects.
I see. Okay.
I have to go to my next meeting, so I will hang up. It was a great meeting. Thank you guys for your time, and please continue without me.
Thanks, Charlie.
Bye.
Bye.
So, the other thing that I want to quickly talk about is the data map testing plan. So we want to have the version 2 released on the 28th of this month. And so we want to have some testing of the UI and the different functionalities to be done starting February 16th. So what I'm looking for are volunteers for the testing. So we will use, as we did earlier, Trello. I've just quickly generated something, but this is not the final form yet. And so whoever tested for us last time are on the card by default. Everybody, Yuling and Huni, Burak, Jeff, all of you, Nansu, Ron, Ian, everybody is on there right now. But if there's anybody else who would like to help us out by testing, please let me know. That would be great. And I will send out an email to everybody about the testing with a link to the testing list, the plan list, as well as on Trello. And each of you, if I get to know ahead of time the people, then I can distribute the testing to the different functionalities that we want to look at.
Forward that link to Yuling so she can
click on it.
Okay. I will. So Yuling, you will have some additional people just like you did last time who will do the testing, and you can just send us a comment.
Yep.
Okay.
All right. So is there anybody else who would like to volunteer?
This is Kathleen. I'd be interested.
All right, Kathleen, I will do that.
Okay, thanks.
Is there anybody else?
Hi, Anu.
Yeah.
So it won't be me, but I know someone who is really interested in testing DataMed. I will connect him to you.
Okay,
separate email.
Okay. Thanks, Eunice. So if you connect us, then I'll put him on there.
Okay.
All right. Okay, I think that's it. So we finished 2 minutes early today.
Okay, great. All right. Thanks, everyone.
Thanks, everybody.
Thank you.
Thank you.